Welcome everyone to the 2019 Net Cancer Day Symposium. We are here hosted by City of Hope. I'm Lindsay, I'm the Director of Communications. And
I'm Lisa Yen, I'm the Program Director.
First of all, thank you so much to City of Hope for hosting, again, this amazing event. Thank you for all of you for joining us and those who are tuning in on the live stream as well. I wanted to remind you of the symposium schedule today. The first speaker we have is Dr. Lee at 11, and then Elyse Gellerman from NetRF will be speaking at 11:30, followed by a Q&A with her. And then we'll have a break at 12:15 for about 10 minutes, and then Dr. John Williams will be speaking as well. And then a big reminder is that we're having, for our first time, a caregiver breakout session during lunch. So the caregivers can stay in this room when we break for lunch at 1:30. And it's about 45 minutes, and then you can go out and get your lunch after. Reminder, no food in the auditorium. And then a few announcements, and then we'll say hello to Giovanna. She wanted to say hi. The 2020 annual Los Angeles Net Conference will be here at City of Hope, June 20th, 2020. So mark your calendars. It's a full-day conference. And then our next LACNETS meeting is on December 10th at UCLA, and that will be with Michael Esselin. And then exciting news, we have the very first wellness retreat for NETs. This is for NET patients living in the Southern California area. It's on December 7th in Beverly Hills. All the information's on our website, lacnets.org, and we take up to 15 patients. So if you want to apply, please apply. It's first come, first serve. Yeah, so there's going to be yoga, a meditation, art therapy, some music therapy, sound healing. It's going to be an amazing day. And then I'll hand it off to Lisa.
I just wanted to echo too, the December meeting with Michael Esselin is going to be excellent. He's a chaplain. He's given TED Talks, and you can check him out online. And I would encourage you to come. It'll also be our holiday celebration. I also wanted to jump in and welcome all of you. I understand we had 100 people RSVP, and we had about 32 people who are attending NET conference or LACNETS meeting for the very first time. So welcome to all of you. Thank you. And since there's many newbies here, I just wanted to mention a couple programs that we have that could be great help to you. One is a patient-physician communication tool that Giovanna and I developed along with Dr. Dan Lee here at City of Hope. It's called Net Vitals, and we have copies on our LACNETS table outside. You can also go to our website, lacnets.com/netvitals for the fillable or printable version. And it's a communication tool. It's a patient-led communication tool where you can go online or go onto the paper and identify areas, the key areas about your disease to help you understand your disease better, so for self-education, and then bring that to your appointments with your physician as a communication tool. It could help identify gaps in knowledge, and then the questions, better ask questions that you have for your physician, and then hopefully then use that time better with your physician. So that's a really great tool we have. We also have a couple programs, including a health coaching program where we provide complimentary one-on-one health coaching services. So more about wellness. So we talk a lot about disease. This is our space for patients or caregivers to individually receive services to talk about how to best thrive while living with disease. And we have health coaches that provide these services. It's done telephonically, and right now it's limited to a certain number of Southern California residents only. So contact me if you have questions about that. Another program we have is Net Connect. So especially for those who are newly diagnosed or early on in those disease, to connect with someone else who's a little further along. And we have several mentors. A few of them are here, and I'll have them stand up so you can identify them, recognize them here. And there's some who are not here. Okay, so there's Val in the back, and Brent and Heather Davis, who's our caregiver. There's several more who are not in attendance, and we have different people in different geographical locations, as well as who are living with disease with different primary tumor origin or even unknown tumor of origin and different symptoms and different treatments. So we can match you with someone based on what you're looking for. That's been a great resource for many people. I also wanted to introduce Mark Sudak, okay, who's going to share with you about an upcoming concert that Marcy, his wife, his lovely wife, who's not here today, is producing and to benefit LACNETS. And as you know, all of these meetings, the monthly meetings, these events like this, that Cancer Day Symposium and the annual conference, as well as all the programs, are made possible by the support of all of our supporters and donors, and every bit counts. So we appreciate all the support and contribution. And for example, we have upcoming Giving Tuesday and just end of year contributions. But this is one specific example of something that is being done to help support LACNETS. So here.
Good morning, and thank you all for what you do. The entire LACNETS effort has helped, I'm sure everyone in this room, I'm speaking personally, it's been a vital addition to my life. Thank you, thank you. If you love classical music, I'd like to call your attention to an event that's taking place 1 week from today at Shoreline Village in Long Beach, basically where the Queen Mary is located. One of the world's foremost violists, Brett Dubner, who performs literally around the world, and we consider a friend, has consented to do a concert, the proceeds from which will benefit LACNETS. 100% of what comes in from this concert goes to this effort. 1 week from today in Shoreline Village, it's an afternoon performance, so it'll be, I think, a little more convenient for us all. Brett will be performing in concert for roughly an hour. There will be a reception afterwards. Your contributions to the event are tax deductible completely. And if I can direct you to a website, particularly for those who are viewing and don't have access to the flyers that are on the table out front. But our organization is called Musique sur la Mer, Music of the Sea, basically. We're located in Shoreline Village, where this event will take place. But if you were to go to the Facebook address for Musique sur la Mer, and I'm going to spell the name for you, it's Musique, M-U-S-I-Q-U-E, sur, S-U-R, la, L-A-M-E-R. If you go to the Facebook page, I was just double checking, the November 7th entry on that timeline is the flyer that's out on the table here. A wonderful concert, world-class performer, and everybody involved in this effort is the beneficiary. So we hope you can come. We have space for a few more, and we need you. And the dollars that come in are obviously needed. So thank you for the time. And I'm looking forward to the afternoon with all of you. Thank you.
Thank you, Mark and Marcy. So I'm going to hand it off right now to Giovanna Mbezi, who is our executive director, as you know, and she's joining us remotely from Northern California.
Hi, can you hear me? Do we have audio? Okay, great. Thank you so much. I'm joining you from Northern California, and I'm just so moved to hear all the extensions of what LACNETS started out as and what it's become over these 7 years, and things like this benefit that Mark and Marcy are doing, and all the programs that Lisa listed, and resources for patients and caregivers, and the event with Michael Esselin coming up on December 10th will be really special, as well as the wellness retreat. So we're really expanding, and it's only thanks to your contributions which make all these programs possible. And I want to thank everyone for the birthday wishes yesterday. That's why I usually take this weekend off to celebrate. And my theme is always age is a privilege. And I'm happy to be older. So I thank everyone. I thank Elyse Gellerman for coming all the way out from Boston to present for us today on the latest research, and Dr. Dan Lee and John Williams City of Hope has been enormously supportive. And I do urge you to check out the Net Vitals document that Lisa mentioned. It's something that's been especially helpful for patients at the beginning of their journey, being able to understand some of the terms, just learn the net language. So I'll turn it back over. I'm looking forward to hearing all the presentations today, and thank you all for coming out, and thank you for your support.
Thank you. We miss you.
Miss you too.
So it's my honor and privilege to introduce Dr. Dan Lee, who's the co-director of the Neuroendocrine Tumor Program here at City of Hope. And he's a huge supporter of NET patients, both here at City of Hope, of LACNETS, and he's become a huge friend as well. On a personal note, as many of you know, my husband lives with neuroendocrine tumor, and I first met Dr. Dan Lee here at City of Hope 3 years ago at a symposium that they were hosting. And he came in with a lot of excitement and dreams and vision, hopes for our community as a whole, which he's really, it's infectious, it's contagious, and he shared along that with us, with Giovanna and I, and our community, encouraged us to dream big and reach for the stars. So thank you for all your support. And here's Dr. Dan Lee.
So I'm going to be discussing with you really clinical trials updates because we really know that clinical trials is, again, how we advance the progress in terms of treatment for patients with neuroendocrine tumors. So these are my disclosures. So in terms of a little bit of background in terms of how we think about drug development and how really things actually get into clinical trials, I think what patients don't oftentimes realize is that this is a really long-term process. So in terms of initially trying to find a drug that might work for neuroendocrine tumors or other types of diseases, scientists all across the world might start with something like 10,000 different types of compounds. But while this is a large amount number in terms of compounds of potential interest, that quickly gets whittled down in terms of the preclinical stage, in terms of testing on mice or other types of preclinical organisms, to approximately about 250 compounds. And then once it's about that 250 compounds that's potentially efficacious in preclinical models, ultimately only about 5 compounds actually make it to clinical trials. And even once those compounds make it to clinical trials, they go through different types of phases within the clinical trials network and process, and ultimately only one drug gets FDA approved at the end of the day. So again, as you can see, it's really a long-term journey in terms of getting a drug initially from drug discovery, ultimately into patients as an FDA approved agent. So we're really going to be focusing on, kind of clinical trials. And as a background, there's various different types of phases of clinical trials that your doctors might tell you about when you're seeing them in the clinic. First, we usually start off with something that's known as a phase 1 clinical trial. So a phase 1 clinical trial is really evaluating the safety of the medication. So we're really looking at, how the drug is ultimately absorbed, distributed, and metabolized in human beings, and really making sure that this is really safe before we take it to the next step. And then the medication will move to the phase 2 clinical trial setting, where the primary endpoint here is really trying to see whether or not the medication is efficacious. Here, we're really getting an additional sense of potential short-term side effects as well as safety, but really trying to investigate a signal in terms of whether or not the medication will work for patients with neuroendocrine tumors. And then finally, if we do see a signal, meaning that the medication is now safe and efficacious, and we have found the dosing that's potentially efficacious for the patients, then the compound will move to what's known as a phase 3 clinical trial. So a phase 3 clinical trial is really now comparing this new medication to the current standard of care treatment to see if whether or not that medication can best that current standard and potentially lead to a new FDA approval. And then ultimately, once a medication is FDA approved, there's something that's known as a phase 4 clinical trial, which oftentimes don't get talked about as much. And this is really what's known as a post-marketing study, meaning that the drug is already now FDA approved, and we're now trying to get more of a real-world experience with the medication in patients, because we know that sometimes for phase 3 clinical trials, while patients are going through those clinical trials, they tend to be very selective patients that enroll on these clinical trials and not necessarily are able to really apply to truly real-world clinical scenarios. So what are some key clinical trial endpoints that you should know whenever you're considering a clinical trial? So I did an extensive review on this topic in terms of various different endpoints previously for LACNETS in 3 years. So I encourage you to look at that video for more detailed analysis. But in general, I would say one of the main key clinical trial endpoints is still overall survival. I would say this is truly the gold standard. This is really trying to define the percentage of patients who are living at a defined period of time. And it's oftentimes reported for neuroendocrine tumors potentially as a 5-year survival rate. What I would say is that you do have to be careful, especially for our disease, really looking at this as a primary endpoint, because we know that neuroendocrine tumor patients will be treated with numerous different types of therapies throughout their neuroendocrine tumor journey. Oftentimes, in terms of the primary endpoint of overall survival, might not be the best initial endpoint, because as you can imagine, if you're looking at a treatment that's trying to get approval in the earlier stage setting or in the initial first-line setting, that's potentially your survival is going to be impacted by all the treatments that you've had later on in their disease treatment course. So that's the caveat that I would give for overall survival. But if anything does show that there's an overall survival benefit, that is actually really powerful, and most likely that agent will be FDA approved. So as a potential surrogate for this, another key clinical trial endpoint that we look at is something that's known as progression-free survival. So progression-free survival is really focusing on the percentage of patients who continue to live with the disease that is stable or better, essentially showing you that the disease is controlled. This is important because we know that as the disease starts to grow, patients can become more symptomatic. And this is potentially an endpoint that is really important to patients, because as long as the tumor is controlled and you're living with the disease, that's okay, because you might be going down the street and someone might be seeing you with a diagnosis of neuroendocrine tumors, but they might not know it because your disease is ultimately well controlled. The advantage of progression-free survival compared to overall survival is the fact that it's not affected by something that's called crossover. What does that mean is that sometimes on clinical trials, when we're testing a drug and we're looking for overall survival, if you're randomized to something that's known as the placebo group, we might allow you to cross over to the experimental drug group and see if it still has an effect. And that can diminish the effect on overall survival of the therapeutic agent that's being tested. But for progression-free survival, because you're only looking at the time until the disease starts growing or controlled, it's not affected by that crossover, because once the disease starts growing, even on that placebo arm, you're able to capture that. So that's one of the advantages of using this as a primary endpoint for certain types of clinical trials. Another secondary endpoint that we often look at is also quality of life. So quality of life, I would say, is very important for this disease, because again, we're really trying to turn this disease into a marathon. And as a result of that, you're going to be living with your disease for a long period of time and living with your disease on an everyday basis. So the therapeutic agent that's being tested in the clinical trial not only have to be efficacious, but you have to be able to tolerate the medication, you have to be able to take the medication on a daily basis, and it should not negatively impact you so much that it's potentially worse than the disease itself, right? So that's absolutely important, and that's oftentimes a secondary endpoint that we assess through various different types of questionnaires or scales in clinical trial setting. Another secondary endpoint is something that's called response rate. So I get this question a lot when patients come to me and they say, what is the response rate of this treatment that you're proposing that I enroll on a clinical trial? So response rate is really based off of something that's called tumor size measurements. We look at your tumors and we really see, how large are the tumors, and see when we test an experimental agent on you, how does that tumor size change over time? But it gets tricky because we use something that's called this RECIST criteria. So this is fancy medical term that tells you in terms of what is classified as a response and what is not. So this really lists how we classify that. So when you get a sense of what the response rates of these agents are, you're not freaking out, because if the response rates are really low, that doesn't mean necessarily that our treatment are not effective. It just doesn't fit the criteria of what's classified as a response in the research setting. So for example, many of you are on first-line treatment with somatostatin analogs. And somatostatin analogs is a great treatment in the sense that it can prolong the disease and keep the disease in check for long periods of time. But the problem is that if you're looking at it from a response perspective, from a patient perspective, and say, what's the response rate of patients to somatostatin analogs based off of the clinical trial language, the response rate is in the single digits, and it's usually less than 5% for patients. But that doesn't mean that it's not an effective treatment. So that's something to keep in mind. So when we look at response rates, if we're quoting you numbers, these are the things that we're talking about. So a complete response is that on imaging, we see that there's absolutely no evidence of measurable tumors. A partial response means that there's a greater than or 30% reduction in the size of all measurable tumors. Stable disease is when it doesn't fall into the category of a partial response or progressive disease. And progressive disease is if there's greater than or equal to 20% increase in the size of tumors. So now with that in mind, when you're looking at response rates are only really capturing those patients that have partial response or complete response in clinical trials. So if that number seems a little bit low to you, look a little bit further in terms of how many patients are actually getting stabilization of their disease and what is the durability of that disease control over time. That might impact your decision in terms of how an experimental agent might be working and might be right for you. So this just summarizes what I just said in terms of objective response rates, which is what is traditionally reported based off of the partial response and complete response. Disease control rates are really important because now it includes those patients with stable disease, and then the duration of response in terms of how long you get control of the disease before the disease grows. And then the final main thing that gets captured on clinical trials is what's known as adverse events. So these are unfavorable symptoms, signs of the disease associated with use of treatment, and there's different types of grading. So this is really telling you what are the potential side effects of the treatment that you're potentially going to be encountering on a clinical trial. A lot of this data will initially come out of the first phase 1 clinical trial of that particular drug, where they're really focusing on safety. So grade 1 is really, mild or no symptoms, and it's fairly well tolerated. Grade 2 meaning that it's, moderate, where oftentimes non-invasive intervention may be indicated. One example of this is usually sometimes nausea and vomiting that does occur with some experimental agents very mildly. Grade 3 is whenever you go to an emergency room or you go to a hospital, that's automatically classified as a grade 3 event, meaning that it's really actually somewhat severe, but you're not potentially in immediate life-threatening situation. And grade 4 is obviously very serious, where it's potentially life-threatening consequences and urgent intervention is required. And then clinical trials will report a grade 5 event, meaning that someone potentially passed away as a result of that agent. And that's something that you have to look at very closely when you're looking at potentially clinical trial data. So before I really dive into some of the kind of key clinical trial updates, this is really kind of the current treatment landscape for neuroendocrine tumors. So what I have summarized for you on this slide are the pivotal phase 3 trials that ultimately led to FDA approvals of various different types of agents that we're using for neuroendocrine tumors. So on the first 2 lines, what you will see is the data that's being presented for somatostatin analogs. Whether you are being treated with octreotide or lanreotide, this is really where the data is coming from. And what you can see is that, again, somatostatin analogs don't necessarily shrink the tumors, but somatostatin analogs have a huge impact in terms of stabilizing the disease and controlling the disease for long periods of time. Oh, yeah. So if you look at, the first two lines. So the first line is what's known as the PERMIT trial. So the PERMIT trial really randomized patients to octreotide versus placebo in patients that had what's known as mid-gut neuroendocrine tumors that were grade 1 or grade 2. And it didn't report any significant shrinkage of the tumor, meaning that there wasn't really much of an objective response rate from the treatment. But what it showed was that there was stable disease in 66.7% of patients. So again, this was really a stabilizing agent for these patients. And in terms of progression-free survival, those patients that got treated with octreotide versus placebo had a benefit of improvement in terms of progression-free survival from 6 months to 14.3 months. Okay. Similarly, in the ClearNet trial that looked at lanreotide versus placebo in patients with advanced gastroenteropancreatic neuroendocrine tumors, again, really not much of a response rate in terms of tumor shrinkage, but in terms of median progression-free survival at the time of the interim analysis for the placebo group was at 18 months, whereas for those that were being treated with lanreotide, it had not been reached yet. Then moving forward in terms of the target therapies, these are what's known as the RADIANT trials. The RADIANT trials looked at a medication called everolimus in patients with either functional neuroendocrine tumors or non-functional neuroendocrine tumors or pancreatic neuroendocrine tumors, depending on the RADIANT trial. So the RADIANT 2 trial was focusing on functional neuroendocrine tumors. The RADIANT 4 trial was focused on non-functional extrapancreatic neuroendocrine tumors. And the RADIANT 3 trial was really focused on advanced pancreatic neuroendocrine tumors. And what you can see also for everolimus is that, again, this is more of a stabilizing agent in terms of response rates. It was anywhere between 2 to 4.8% in terms of 30% or more shrinkage of the tumors. But in terms of median progression-free survival from the trials, at least for pancreatic neuroendocrine tumors, as well as extrapancreatic neuroendocrine tumors that were non-functional, it did lead to a significant median progression-free survival benefit compared to placebo. Similarly, there is a medication called sunitinib for patients with pancreatic neuroendocrine tumors that was also tested in the phase 3 setting. Again, still more of a stabilizing agent in terms of response rates of only 9.3%. But again, similarly to everolimus, it led to improvement in progression-free survival from 5.5 months to 11.4 months for patients with pancreatic neuroendocrine tumors. And then, everyone is obviously really focused on PRRT with lutetium-177. So this was the NETTER-1 trial, which is on the last line here, that really focused on patients with advanced well-differentiated mid-gut neuroendocrine tumors. Here now we're getting additional signal in terms of tumor shrinkage. So response rates that were reported in the original NETTER-1 trial approach 18% for those patients that were getting treated with PRRT. So again, that's not including the patients that had stabilization of the disease. So while most patients had stabilization of disease, now you're getting into an area where you're getting double-digit, potential significant shrinkage of the tumor of 30% or more compared to octreotide alone with a higher dose of octreotide of only about 3%. And the median progression-free survival was not yet reached at the interim analysis for those patients that were treated with PRRT compared to the higher dose of the octreotide, which was a median of 8.4 months. And overall survival, while you see that the curves are actually starting to separate from the clinical trials, from the NETTER-1 trial, we do need additional time because as I told you, for this disease, oftentimes we need to potentially get to 5 years before we see those curves potentially able to fully separate to say that whether or not we have a potential survival advantage in terms of overall survival. This is kind of also adding to that in terms of treatment with chemotherapy. So we don't oftentimes talk about chemotherapy for most neuroendocrine tumors, with the exception of, I would say, pancreatic neuroendocrine tumors, and then also potentially with some lung neuroendocrine tumors. And what you can see is that this is really retrospective analysis of patients that were being treated with chemotherapy for their neuroendocrine tumors and predominantly pancreatic neuroendocrine tumors. But what you see is that in terms of response rates, again, if you're really trying to go for tumor shrinkage and you need quick tumor shrinkage of your tumors, with chemotherapy, you're getting a much higher response rate compared to some of the other target agents that we have, whether it's somatostatin analogs, everolimus, sunitinib, or PRRT. And this was actually confirmed in what's known as a phase 2 randomized controlled trial. Again, as I mentioned, a phase 2 trial is really trying to assess for efficacy of the medication. And what this phase 2 trial that was done by the ECOG cooperative group led by Dr. Coons from Stanford was trying to look at a medication called temozolomide, which we know is an alkylating agent and has activity for patients with pancreatic neuroendocrine tumors, and combine it with another pill called capecitabine to see whether or not there's an increase in response as well as benefit with treatment. So this took patients only with pancreatic neuroendocrine tumors, and the primary endpoint was progression-free survival. And what they found was that when the two medications were combined, the progression-free survival, the median was at 22.7 months, whereas if you just took the temozolomide medication by itself, it was 14.4 months. And median overall survival was not reached. And in terms of response rates, I can tell you from this trial, it was consistent to other chemotherapy agents that we use for pancreatic neuroendocrine tumors, where the response rates generally was closer to 30%. So for pancreatic neuroendocrine tumors, if you're looking for quick shrinkage of the tumors, chemotherapy is oftentimes something that we go to for that because of the increased response rate compared to the other agents that we have for this disease, including Lutathera. So this is kind of the general algorithm in terms of how we think about the current treatment landscape for patients with neuroendocrine tumors. So if I would say if you have a gastrointestinal neuroendocrine tumor, really your treatment options are somatostatin analogs, predominantly as first-line treatment based off of a good safety as well as tolerability standpoint, excellent quality of life perspective, limited off-target effects, as well as other agents such as everolimus, which I showed you based off of the RADIANT-4 trial, which is an oral target agent, stabilizing agent, and then Lutathera PRRT. For patients with pancreatic neuroendocrine tumors, we have much more options, whether it's somatostatin analogs, target therapies with everolimus or sunitinib that I mentioned, PRRT, as well as chemotherapy that I told you about if you really need significant shrinkage of the tumors. And then for lung neuroendocrine tumors, really we have everolimus based off of the RADIANT-4 clinical trial that's FDA approved. Somatostatin analogs we do use, although if you were really astute, you might have picked up from my slides earlier that none of the trials that included patients treated with somatostatin analogs earlier on, the phase 3 trials actually included patients with lung neuroendocrine tumors. But there is an ongoing trial called the SPINET trial that is looking at that. And we think that in the neuroendocrine tumor community, we make the assumption that if you have somatostatin receptor positive disease on a gallium scan or the old-fashioned octreotide scan, that you're probably going to potentially benefit from somatostatin analogs, and that's why it's shown here. So we don't have phase 3 data yet, but we are fairly confident that this is an agent that works for you. And then there's chemotherapy, and I put the question mark in terms of Lutathera PRRT because we think that, again, just like the somatostatin analog therapies, while the initial clinical trials from the NETTER-1 trial did not include patients with lung neuroendocrine tumors, we do know that from the European experience, as well as several retrospective studies that have been done, that PRRT probably has a role and potentially has an indication for lung neuroendocrine tumors moving forward. All right, so what is potentially coming in terms of clinical trials updates and agents that are being explored right now in clinical trials or potentially are going to come really fast. I cannot go over all of the trials with you today because there's so many things that are going on in the neuroendocrine community right now. But I will try my best in terms of highlighting some of the key areas I think are potentially exciting and potentially are closest to getting into that phase 3 setting where potentially they can be approved for patients all over the world. So I would say one class of medications that's heavily being looked at right now for neuroendocrine tumors is this group of medications called tyrosine kinase inhibitors. And these are medications that, just like sunitinib is a tyrosine kinase inhibitor that's already FDA approved for pancreatic neuroendocrine tumors, they often target a pathway in cancer growth, something that's known as the VEGF pathway. So this is a pathway that's oftentimes involved in angiogenesis or blood vessel formation within the tumor. And we know that neuroendocrine tumors tend to have a lot of blood vessel formation. And as a result of that, if you're able to target that and inhibit that, you might be able to inhibit the overall growth of these types of tumors. Now, There's several different agents I will cover because I really think that this is going to be what's known as a race between all of these tyrosine kinase inhibitors in terms of who gets the first indication for neuroendocrine tumors, and how do you choose these will be somewhat very difficult for practitioners moving forward if all of these become FDA approved. In addition, I will be touching a little bit on the role of immunotherapy because I know immunotherapy is a hot topic in oncology, and really review the data in terms of single agent immune checkpoint inhibitors, as well as combination immune checkpoint inhibitors, as well as bispecific antibodies. And then give you a little bit update on PRRT, really focusing on potentially trying to improve on PRRT with a somatostatin receptor antagonist molecule. And there are other trials that are being explored, whether it's with alpha emitters, which is a different type of radiation that's being dosed for PRRT, because the benefit of PRRT is that you can always either change the peptide fragment or the radiation molecule, and you're potentially able to possibly improve response and efficacy of the treatment. There is also an ongoing clinical trial that's looking at essentially comparing PRRT to the oral target treatment that we already have in terms of everolimus to address whether or not this is something that's called a sequencing question, meaning that should patients really have everolimus first, or should they have PRRT first, and get a sense of which agent is truly better in that sense. And then there is also an Australian group that's really focused on kind of combination therapies with PRRT. So they're actually combining the capecitabine and temozolomide that I showed you data earlier for pancreatic neuroendocrine tumors, and combining it with PRRT to see whether or not that will give you an even more increase in response rates compared to just chemotherapy alone, maybe taking it from 30% in terms of response rates with chemotherapy to possibly 50%, and whether or not that's a synergistic property of the different types of medications or additive. So first, with the tyrosine kinase inhibitors, so this is data that was initially presented in 2017 focusing on cabozantinib. So this was a study that was presented by the Dana-Farber group, which took patients with various different types of well-differentiated lower intermediate grade neuroendocrine tumors, and they were allowed to have any type of prior anti-VEGF treatment, so any of those other VEGF compounds such as sunitinib, and then they were treated open label with cabozantinib until their disease started to grow or until unacceptable toxicity, and their primary endpoint was response rate. This is something that we call a waterfall plot. So waterfall plots are a good way to conceptually look at what the response of treatments are to the experimental agent. And what you can see is that while the response rate was only 15%, meaning those patients that achieved a partial response or better, if you look at the waterfall plot, you see that most of these patients had tumor shrinkage for their tumors if they had a pancreatic neuroendocrine tumor that was treated with this agent. And then for extrapancreatic neuroendocrine tumors, oftentimes we call these carcinoid tumors. Again, while the response rate was only 15%, many of these patients did have tumor shrinkage. And as a result of that, this molecule is now being tested in what's known as the phase 3 setting in the Alliance Cooperative Group, where it's taking patients that previously had everolimus for either pancreatic neuroendocrine tumors or extrapancreatic neuroendocrine tumors, and it is randomizing patients 2-to-1 to either cabozantinib or placebo. And it's really trying to show whether or not cabozantinib is a potential efficacious agent for neuroendocrine tumors moving forward. So the next tyrosine kinase inhibitor that I'll hit on is this medication called lenvatinib. So lenvatinib was being looked at by the Spanish group, and the Spanish group did a phase 2 trial to really, again, look at the efficacy of lenvatinib in patients with neuroendocrine tumors. And these are patients, again, with, they included a cohort of patients that had pancreatic neuroendocrine tumors, as well as neuroendocrine tumors of the gastrointestinal tract. The lenvatinib dosing that I would say was 24 milligrams, and as a GI oncologist, I can tell you that this is sometimes a little bit higher dosing than what we usually do for other types of GI cancers such as primary liver cancer, which is a much lower dosing. So potentially there's a possibility of increased side effects with this dosing. But what's remarkable about this agent is that if you look at pancreatic neuroendocrine tumors, now you're seeing potentially response rates of 40% with an oral pill. This has really not been encountered ever with an oral medication. So this medication at this dose is showing significant tumor shrinkage for a majority of patients. And again, as you can see on this waterfall plot, most of the patients were benefiting from treatment in terms of tumor shrinkage or not. And even for extrapancreatic neuroendocrine tumors, now we're getting into response rates of 18.5% with a majority of patients showing tumor shrinkage. So again, this medication, I think, is a very interesting molecule. And we have seen it as an effective molecule for other types of diseases that really rely on blood vessel formation, such as kidney cancer, as well as primary liver cancer. And I think neuroendocrine tumors are potentially a really good tumor type to potentially benefit from this agent. So the question is whether or not, as a community, we want to move forward with a phase 3 trial of looking at lenvatinib versus either a different agent or versus placebo to see if we can see a positive effect. There's this other medication called pazopanib, which was also a VEGF inhibitor that was presented by Dr. Berglund from UCSF this year at ASCO. So these were patients that had extrapancreatic carcinoma tumors, where normally we don't use agents such as VEGF inhibition, and they were randomized to pazopanib versus placebo. They did allow, this is what I'm talking about in terms of crossover from earlier, they did allow those patients that were randomized to placebo to cross over to open label pazopanib if they developed initial progression. And as a result of that, the primary endpoint was really looking at progression-free survival. As I mentioned before, progression-free survival is not impacted by crossover, so that's how you are allowed to do this study. So what you can see in terms of the primary endpoint was that the median progression-free survival for placebo group was 8.5 months, and for the pazopanib group was 11.6 months. Now, I would say this is, while this was statistically significant, the benefits in terms of progression-free survival was relatively modest. As I showed you previously from the RADIANT trials, oftentimes you're getting an improvement in median progression-free survival of potentially 5 months or more with everolimus or sunitinib. And now you have an agent that's only improving your progression-free survival of about 3.1 months. So while this was a phase 2 randomized study that did meet its primary endpoint, the question that I think we have to ask within the community is whether or not this is clinically meaningful for us to really take this to the next level and potentially do a phase 3 study to lead to its FDA approval. In addition, this medication is somewhat toxic. It does have a lot of issues in terms of having high blood pressure and other side effects. And as a result of that, if you look at the trial data from this study a little bit deeper, many patients require dose reductions. Many patients require being taken off of the medication within the trial. So that's something to, again, keep in mind from the quality of life perspective. And then finally, the most exciting thing that came out was with this VEGF inhibitor, and I would say this is probably potentially at farthest along in terms of development of all of the VEGF tyrosine kinase inhibitors. So this is a medication called sulfatinib. Its name has been changed to surufatinib. And this was initially presented at the European Neuroendocrine Tumor Society Conference in 2017, looking at sulfatinib as a single agent for patients with pancreatic as well as non-pancreatic neuroendocrine tumors and continued until disease progression or toxicity, with primary endpoint being response and safety. Same thing in terms of a waterfall plot, very encouraging waterfall plot for the pancreatic neuroendocrine tumors, response of 17.1%, but overall disease control rate of greater than 90% in these patients with pancreatic neuroendocrine tumors. Same thing for extrapancreatic neuroendocrine tumors, about 15% response rate and disease control rate of 92.5%. Now, as a result of that, this is a molecule that's being developed in China. So for the Chinese patients, they already did a phase 3 clinical trial that randomized patients that had extrapancreatic neuroendocrine tumors 2-to-1 to surufatinib versus placebo, where the primary endpoint was investigator-assessed progression-free survival. So this was just presented this year at the ESMO Congress. And what you can see is that the medication did meet its primary endpoint in the phase 3 setting for improvement in median progression-free survival from 3.8 months to 9.2 months. Now, this is a little bit tricky in terms of how we get this drug somewhat approved going forward, because I would say this is the first drug where we did the phase 3 in China and not necessarily in the United States or Europe. And how is the FDA as well as the European regulatory office going to look at this data and translate it to the Western population? We do have an ongoing phase 1 study that's looking at the safety and tolerability of this compound in the Western population, and that has completed accrual. I think there might be 2 more spots that might be open, but I think it's pretty close to accrual. And we have found that this is really the toxicities that have been reported by the Chinese group are really similar to the Western population. So I think it will be, again, interesting to say, is that enough to get the FDA approval here and really bring a new treatment option for patients that have extrapancreatic neuroendocrine tumors, where really you only have 3 lines of FDA-approved treatments, or whether or not we have to repeat the study as a community and do another phase 3 study in the Western population to see whether or not this is an efficacious compound. These are just secondary endpoints, and what you can see is that for many of these patients, they did encounter some form of tumor shrinkage from the medication. So what about immunotherapy? So I think in general, I would say that my general comment for immunotherapy is that we had a lot of investment in immunotherapy. We had a lot of hopes for immunotherapy. I don't think we're there yet in terms of immunotherapy. So this was presented at ESMO last year, and this was looking at an immunotherapy drug called spartalizumab in patients with various different types of neuroendocrine tumors, whether it's pancreas, gastrointestinal, lung, as well as high-grade neuroendocrine carcinomas, for which we participated in this study. So the idea and the rationale behind immune checkpoint inhibitors is that what it does is that these immune checkpoint inhibitors block a pathway that's involved in terms of a tumor escape known as the PD-1 or PD-L1 pathway. It's a way for potentially your tumors to evade its sense from your T cells that circulate throughout your entire body. And if you give an immune checkpoint inhibitor, it basically blocks that pathway that allows the tumor to not be recognized by your T cells. And previously, there was a study of another immune checkpoint inhibitor called pembrolizumab that demonstrated a response rate of 6 to 12% in patients with pancreatic neuroendocrine tumors as well as advanced carcinoid tumors. So we thought that it was a good idea to then look at this in patients with different types of neuroendocrine tumors. So this was this phase 2 study design, which included patients from GI neuroendocrine tumors, pancreatic neuroendocrine tumors, thoracic neuroendocrine tumors, and then also high-grade, poorly differentiated neuroendocrine carcinomas. And the primary endpoint was response. And in summary, what I want to show you on this table is that really it didn't work. All right. If you look at response rates, again, greater than 30% or shrinkage of the tumors, there was not much of effect in GI neuroendocrine tumors, not much of effect in pancreatic neuroendocrine tumors, really not much of effect in terms of poorly differentiated neuroendocrine carcinomas. And then interestingly, for the lung group, there was an effect. But what we realized when we looked at subgroup analysis is that the only patients that developed a response in the lung group were these patients that had what's known as atypical carcinoid tumors of the lung, or a little bit higher grade, not quite high grade lung neuroendocrine carcinomas, but kind of that intermediate, a little bit more aggressive lung neuroendocrine tumor. So as a result of that, this study is not going to go forward as a phase 3 study because we did not really see much of a signal from the phase 2 efficacy endpoints. And these were response rates that were shown for all the different treatment groups. Similarly, Dr. Strasberg also presented this earlier at the GI ASCO in January this year with a medication called pembrolizumab, which is really just like spartalizumab as a phase 2 study for patients with various different types of neuroendocrine tumors. And just like the study that we did, really showed not much of a response rates in patients with neuroendocrine tumors. And they even looked at it a little bit deeper in terms of biomarker analysis, saying that if they stain your tumor to show something called PD-L1, which is that pathway that I'm talking about, that this molecule is potentially targeting, will that potentially lead to an increase in response? And what you can see is that there was actually really no difference between the PD-L1 positive tumors compared to the PD-L1 negative tumors. And in fact, all the PD-L1 negative tumors were the people that actually responded to treatment, which tells us that we're really not as smart as we think we are with immunotherapy, and that more needs to be done. So where are we going with this? I think we're moving a little bit forward. So this is where potentially bispecific antibodies come in. So this is a different form of immunotherapy. So how bispecific antibodies work is that on the one end of the molecule, they're trying to target a peptide or a protein. And we obviously in the neuroendocrine tumor world have the advantage that we can potentially target somatostatin receptors, right? Because a lot of our tumors have somatostatin. So the idea is that if we can target that, and then on the other end of the molecule, there's this molecule called anti-CD3, can we then, essentially migrate the T cells directly to the neuroendocrine tumors? Instead of blocking a pathway of resistance, can we really try to force the T cells to go towards the neuroendocrine tumors and allow the immune system to attack the neuroendocrine tumors in this fashion? So this is a study that's in the phase 1 setting. Again, really assessing safety and tolerability, and as well as finding the dose. And us, along with several other institutions, are involved within this study. So hopefully this will shed light in terms of the future role of immunotherapy moving forward. What about also, what are we doing from a PRRT perspective? So again, I think Lutathera has been a really encouraging agent, response rate of 18%, great long-term durability control with long progression-free survival and potentially overall survival benefit. But just like other investigators, we're always thinking in terms of how can we do better? So this is where potentially somatostatin, targeting somatostatin receptors with something that's known as antagonists might benefit. So what's the difference? The difference is that if you use a somatostatin antagonist compared to Lutathera, which is on the peptide side of the molecule of Lutathera, is what's known as a somatostatin agonist, is that the antagonist may have the ability to bind independently of somatostatin receptor activation states. So potentially you could possibly get a little bit tighter of binding with the agent. And the idea is that if you have a little bit of tighter binding, would you be able to potentially occupy more binding sites on the tumor surface? And as a result of that, increase the efficacy of this medication. So we're participating in this phase 1 trial along with MD Anderson, as well as many European colleagues that's looking at, how can we use this somatostatin receptor antagonist molecule called lutetium-177 or OPS-201. The other name for it is called JR11 in terms of seeing whether or not this will give us a little bit tighter binding on patients with neuroendocrine tumors and then potentially lead to overall better efficacy. So I know that's, kind of quite a lot and somewhat of a whirlwind, and I probably took way too much time, but, hopefully I've given you some sense in terms of I think the future is really bright for all of us. There's a lot of molecules that we're looking at. It's really trying to decide which are the best molecules to take to the phase 3 setting so that ultimately we can potentially get quick FDA approval so that it will be in our arsenal to treat all of you with these novel compounds. Thank you so much. So I think we'll save questions to the end. So I'm going to introduce Elyse Gellerman. So Elyse is the Chief Executive Officer of the Neuroendocrine Tumor Research Foundation, positions that she has held since November 2017. She joined NetRF in September 2016 as a Senior Administrative Officer. And Elyse has done great things with NetRF, and I think she'll be able to highlight some of the translational work that they're doing, some of the more preclinical stuff, and ultimately translate it for us quickly in the clinical setting. Elyse.
Good morning, everyone. It's my pleasure to be here with you. I want to thank LACNETS for the invitation. It is really a pleasure to spend World Net Cancer Awareness Day with all of you. For you, as well as for us, every day is Net Cancer Awareness Day. So we're really enjoying the opportunity not only to present and talk to you a little bit about the work that we do, but also to hear some of your stories and get some feedback so that we can better provide resources and education to all of you. In terms of LACNETS, I just want to congratulate the group on everything that it's been doing, the Net Vitals, making the meetings available to everyone through a live stream. I think LACNETS is one of the groups that truly transcends its geography. So we really appreciate everything that the group does for the NET community. Okay, I'm trying to use the okay, we'll go low tech. All right, so I'd like to talk to you about three things in my time today. Some fast facts about NetRF so you understand a little bit about who we are and what we do. I'm going to talk about some general trends in research. As Dr. Lee said, if you think about the process of developing treatments as a continuum, a lot of what we do is at the early stage. A lot of what we fund is in that discovery phase. And then I just want to end with a couple of notes about patient advocacy and opportunities to be involved in research. I always like to start with a story. The Net Research Foundation was established in 2005. And we started because a patient was diagnosed out of the blue, a situation I'm sure a lot of you can empathize with. And she collapsed in the Boston subway one day. She was a professional woman in Boston. She was taken to a local hospital and she was diagnosed with a neuroendocrine tumor. She had never heard of neuroendocrine tumors. She asked about treatment options and really was told that was quite limited. Remember, this is about 2005. And she went home, and with that same focus that she applied to her work, she started to research what was available for neuroendocrine tumor patients and look at who was doing research. And back then, she found very little. So she put together a group of patients and family members in the Boston area who had common interest and experience, and that became what was first called Caring for Carcinoid. If any of you have been on this journey for many years, you've probably heard about Caring for Carcinoid. And she also pulled together a panel of some of the very smart people in science in the Boston area to form a board of scientific advisors. Some of those people continue to work with us today, and we're very grateful for that. So we became Neuroendocrine Tumor Research Foundation in 2015. We rebranded ourselves to really focus and communicate to everyone what our mission is. Our mission is to fund scientific research, to discover cures, better treatments, and also to provide educational resources to patients as they're on this journey. We've been true to that mission since 2005, and we have grown significantly over the past couple of years. So we're very pleased that our mission has certainly resonated with a lot of other people. So we now have a global reach. We've funded more than $22 million in research. Since we started, we'll probably pass $25 million by the end of this year. And that really translates out to more than 60 projects since the beginning, now on 3 continents. So we want to fund very strategic, innovative work so that we can understand more of the why. Dr. Lee talked about the trials that try to measure response and look at if something is an effective treatment. A lot of the work that we do is basic or translational science to really get into the whys and understand tumors at a very basic level. So we want to understand the biology of tumors, and if you talk with some of our scientific advisors, they'll say until we can fully understand these tumors, we really cannot develop as many effective treatments. As all of you know, these tumors are very heterogeneous. They are different. Your symptoms are different, and your symptoms may be different from someone with the same tumor site sitting next to you. So that makes this a very, I think, profound scientific challenge. We've also been very active over the last number of years in trying to recruit scientists who have been successful and have expertise in other areas in science as well as in oncology, and ask them to start thinking about NETs, because the more ideas we can bring, the more expertise we can bring. This is a wonderful scientific challenge, and I think we've been able to recruit a lot of very bright people to start to make their career in neuroendocrine tumors. So we really want to understand the characteristics of pancreatic and intestinal and lung and liver tumors. It's also important to us to understand why some patients respond and others do not. So again, more of the why around some of these clinical trials. And what we call basic science is scientists in the laboratory with cells, with animal models. So this is very painstaking, very, very long duration work. The translational science is some of those laboratory experiments that have the potential within maybe a 3 to 5 year window to translate into how we treat patients and the type of treatments that we have available in the clinics and the clinical trials. The other thing that's a challenge in neuroendocrine cancer is creating tumor cell models. So cell models are those resources and multiplying cells that scientists can use in their experiment. And we don't have enough of those in neuroendocrine tumors, because what is advantageous in a patient, these tumors grow slowly, or sometimes they just sit, sometimes they're dormant. Not always good in the laboratory, when what you want to do is you want to get those cells to multiply and to be available for scientists throughout the world to use for their experiments. So we're very active in trying to fund approaches to cell models, and we'll talk about that in a moment or so. As Dr. Lee said, the process of discovering and approving a new treatment can run for 15 years or so. A couple of studies say the development of a new drug can be $2 to $3 billion. And what we've always focused on, and kind of our sweet spot, is that discovery period, the first 2 to 10 years, when we learn something on a cellular level and we start to look at some of the agents or some of the approaches that may lead to a new compound. The other thing that's happening in science right now is there is a lot of discussion, a lot of work on repurposing or drug screening or drug discovery, where we're looking at, can we take cell lines or cell models of neuroendocrine tumors and test some of the other thousands of compounds that are out there that have been approved and discovered for other ailments. And sometimes you get a hit on some of those, but again, we're trying to develop cell lines so we have the resource and the cells to do some of that screening. So I want to talk about some of the kind of important trends in research, and Dr. Lee covered some of them, but I'm going to use a framework that was presented by Dr. Robert Jensen, who works with the NIH down in Washington, D.C. He presented this at the LACNETS meeting, which is the North American Neuroendocrine Tumor Society. It's a group of physicians and researchers, and they have an annual meeting. It was in Boston last month, and he presented what I think was a really good list of some of the main areas of interest for scientific research. And I'll go through those, and what I will do is I will illustrate some of those with some of the projects that we are funding, so that you can understand some of that science at a kind of basic level, and really know where people are looking to try and find some of the answers to the questions we still have about neuroendocrine tumors. I'll also give you the takeaway right up front. The takeaway is that really across the world, and as Dr. Lee talked about a little bit, there are so many researchers who are looking into neuroendocrine tumors, and you don't have the opportunity to see them. I'm very fortunate that I have the opportunity to travel around and meet many of these researchers and see their eyes light up when they talk about their work, even though I sometimes only understand 50% of what they're saying. But just know that since you can see the patient community here and in other places that you travel, you are not alone, and there are so many people who are working on your behalf in those laboratories and getting up late at night to go in and tend to their experiments. So know that we have a lot of very smart people here at City of Hope and the Los Angeles area and really throughout the world who are trying to develop better treatments for all of you. And I think that's a really hopeful message, especially on Neck Cancer Awareness Day, that there's a lot going on, and physicians have certainly told me that there are more tools in the toolbox than we've ever had, and more in the pipeline. So, so know that as we go through some of the kind of basic trends. Organoids and cell models, as I mentioned, the cell models, this is really fundamental to a lot of laboratory research. You have to have a model of a tumor, of a disease, in order to really effectively study it and study it in many different ways. So organoids have been developed and have attracted a lot of very good attention in the area. If you think of an organoid as kind of a mini organ, and you'll hear that term, they're three-dimensional structures, so they're grown from tumor cells in a particular way, and those little clusters of cells will mimic the tumors in terms of their function and some of their cellular characteristics. So this is a picture of an organoid. This was a recent issue of Science magazine, and it's another way to try and create a resource for researchers across the world. In 2017, Net Research Foundation funded a grant with Hans Cleaver's lab in the Netherlands. He is regarded as one of the world experts on growing organoids. And that was a good example of where someone who had this incredible expertise had never really thought about applying it to neuroendocrine tumors. And some discussions ensued. He applied for a grant, and we were able to fund that. So we're about halfway through now, and that is looking at trying to create intestinal, pancreatic, and actually some lung organoids. There was a result of that work, some preliminary results presented at a European meeting last year, and also I believe it may have been ASCO or one of the American meetings, and they are showing some response. The response to be able to grow these organoids differs by tumor site, and again, we're about halfway through, so they want to make sure that these organoids continue to grow, that they don't grow to a certain point and stop, which has been one of the challenges with cell lines in the past. So this is, again, another approach to how we can get models to really test treatments and understand what's happening with these tumors as they grow and spread. So if we can have some stable organoids, then we also have the opportunity to expose those organoids to some of the different drugs that are out and see where we get a response or where we don't. But again, it's not in patients, it's at a very basic cellular level. And it would take a long time in that discovery period or that funnel that Dr. Lee presented to really get to the point of a patient clinical trial. So we want to know what are the existing drugs that potentially might be helpful for different types of neuroendocrine tumors. Molecular studies, another thing cited by Dr. Jensen for the progress there. And this is, again, kind of understanding what's the difference in cells, what's happening in the basic biology. And one of the, I think, new areas of inquiry there is subtypes in non-functional peanuts. So this was actually work by Dr. Ramesh Shivdasani, whose picture you can see there, and another lab that's run by Dr. Bradley Bernstein. This work was funded in about 2011, 2012, and it was just published in July. So you can see there was a long lead time. Science has to build upon itself, so it takes a very long time to have these cells grow, to identify something that was previously unknown, to continue to look at those patients. And the interesting thing here in this preliminary work is that in these non-functional pancreatic neuroendocrine tumors, they found that they were divided really into two subgroups that had different types of cells and different proteins, alpha cells and beta cells. And when they looked at those groups and the patients who had donated these tumor tissues, they found that those patients had a different risk of recurrence going forward and looking at those patients over a number of years. So again, this was work in the laboratory, but it was enough of a discovery to be published in a peer-reviewed journal back in July. This work is continuing because if you think about it and you think about what may be the impact in a number of years, if these patients all had non-functional pancreatic tumors, but they had a different risk of recurrence because of these alpha versus beta cells, then potentially this could lead to different paths of surveillance and monitoring of those patients. So we're not there yet, but again, it's an interesting discovery that the researchers are continuing to follow, and it shows you how something that is studied with a lot of specificity in a laboratory, in a dish, can potentially lead to those improvements in not just the treatments, but also surveillance and what we know about the course of some of these tumors over a period of a number of years. We've also, through some of the molecular studies that we've supported over the years, identified the role of MEN1 in intestinal tumors. We have funded work that has looked at mutation in DAX and Atrix genes. Some of those mutations are there, but we don't completely understand the mechanism and the impact. So this has been studied in a number of projects over a number of years. Remember, except for a couple of genetic syndromes, there's no one mutation that people can point to in neuroendocrine tumor patients. But again, the interaction of genes and something called telomeres, it's very complex, but we've seen a lot of progress in understanding this over the years, so we continue to fund some of this work. And hopefully we'll be able to know a lot more as some of our researchers collaborate. That's another big value that we have. We want to make sure that our researchers are collaborating, that they're presenting to their peers, and providing progress reports to us so that we can connect the scientific community so that we get more perspectives, more very smart people who are trying to understand some of these processes. And we're now probably at the point that we have about 50 scientific papers in journals that have been the result of the work that we've funded. So again, we're moving along. We don't know as much as we need to, but there is steady progress. We're also trying to figure out what is the cell of origin, or how do these tumors start in the first place? And looking at some of the stem cells, looking at some of the tumor samples from a variety of different ways. We have an international team in London, Boston, and San Francisco that's looking at some of these genetic mutations to figure out, first of all, are they inherited or are they acquired? Are they just happenstance? And is it possible that they result from something in the cell environment or in the general environment? So there's a lot of work to try to figure out, why do these tumors start in the first place? And what can we do to, if we understand that process, to interrupt it? Imaging improvements, of course, the gallium scan is a wonderful tool that certainly is very effective for a lot of patients, but not every patient. So we have supported some projects to look at amino acids as potentially a different type of a tracer, because not every patient has a somatostatin receptor. So if we can start to design some of the imaging agents that will make scans more effective, pick up tumors earlier and smaller, then that gives us kind of more in the diagnostic arena. We're also now supporting a project in the Netherlands that's looking at photodynamic therapy. And again, it's in the laboratory, but trying to look at a photosynthesizer, a molecule that if you hit it with light, it will light up and potentially cause cell death. Or the other possible application to this down the line would be, if you can get tumor cells to light up, then if you are having surgery to remove a tumor, there is a better chance that that surgeon can visualize that tumor in a different additional way to make sure that all of the cells are taken out. PRRT, of course, it's such an exciting area here since the approval in the United States in 2018, and obviously the availability in Europe 20 years before that. But we have a lot invested in trying to make this therapy more effective and more applicable to many more patients. And Dr. Lee talked a little bit about this. I think you actually mentioned Dr. Hicks's project in Melbourne. So that is a project we support in Australia. The biology behind that is also trying to understand the cell repair process. Why is it that you can hit tumor cells with radiation? Some of them die as you want them to, and some of them have the ability to repair themselves. So what's happening, and if we can understand that repair process, again, there is the potential to interrupt it and make sure that you're hitting all of those cells and that you're causing those cells to die and getting a better response. We're also looking at some of those different radioactive particles, and there's a team in Switzerland who is looking at an alpha particle, because Dr. Lee explained you have two pieces to the PRRT. You have kind of the delivery system, and then you have the radioactive molecule. If you tinker with either of those, can you come up with a treatment which is going to be longer lasting? Or in this case, this particular team is trying to hunt down those micro metastases. So think about it as a couple little cells that get out of the box, and hunting down those cells so that the PRRT can be a little bit more specific, and go out and hunt down some of the cells that might have gotten away to prevent rather some of the metastases down the line. So we're funding that work. And then, as you know, PRRT is not approved for lung. So we are doing actually our first dedicated lung research project, and there will be more of those to come. But a team at Moffitt in Florida is looking at alpha particle PRRT for lung nets. So I think that will be exciting. We expanded our portfolio to cover lung nets over the last year. And as I say, we will have more projects on that probably very shortly. We're just at the tail end now of our 2019 request for applications. We do peer review of all of our research applications, and we'll probably have some decisions and announcements on new grantees at the end of the year. So I want to make one or two additional comments about immunotherapy. Absolutely agree with Dr. Lee. Haven't quite found the bullet with that yet. Not exactly ready for prime time in neuroendocrine tumors. We have been funding work on CAR T therapy, and if some of you are familiar with that, it's been very effective, was developed by a number of people, including Dr. Carl June at the University of Pennsylvania, and was approved for leukemia and now lymphoma. So it's proven very effective in blood. And following that, the question was, can we apply it to solid tumors? What's different in solid tumors? Can this be a way to engineer T cells to really go after neuroendocrine tumors? Again, it's still in the laboratory, but we've seen some progress over the last couple of years, so we continue to fund this work. We are hopeful, but there's I think a long way to go before we get something that would be ready for a clinical trial. The other interesting project we're funding now is a phase 1 trial at Roswell Park in Buffalo. This is actually a vaccine, and it targets a substance called survivin. This was a discovery that they made. They were actually studying brain cancer patients, and what they found was a lot of these brain cancer patients expressed a substance called survivin. And when they just happened to test some patients and some tissue samples with neuroendocrine tumors, they found that almost half of the samples that they had, the neuroendocrine tumor patients expressed survivin on their tumors. So they said, well, let's try a phase 1 trial. So there's about 10 patients that they're in the process of accruing to this trial. And let's see if this vaccine, which is given in a shot over a period of a couple of months, let's see if this has any impact on those patients who had progressed after some standard treatment. So it's an interesting kind of immunotherapy vaccine approach. And it's very early, but we'll see what the response is for those 10 patients or so. The other work that we fund, because frankly no one else will fund it, is something called correlative work. So when you have a clinical trial, as you may know if you've ever been in a clinical trial, there's a very specific protocol. There's very specific blood samples, tissue samples, scans that you need to get as part of this clinical trial. And that's what's studied, and that's what produces the data that Dr. Lee presented, and a lot of the response rates are calculated off of that. And many times after the clinical trial, those samples and a lot of that data then sits. So if you can look at the information that's gathered in that trial and look at it from a slightly different perspective, or try to find out a little bit more at a different level, then we can sometimes learn some things. So we are supporting some correlative work with a study that Dr. Emily Bergslin has been involved in, and Dr. Lee mentioned that. So she's trying to look at evaluation of the blood proteins in those patients who were in the trial, and also do some computational analysis of CT scans. And really the goal of that is to see, can we develop better methods of evaluating and monitoring patients in those trials for their response. So this is really interesting work. Again, the path to drug approval is very specific, and a lot of times the additional information and the knowledge you can gain from these clinical trials is not pursued because it's not part of the purview of that clinical trial and that drug approval and pharma's course for that potential drug. So, we think that that's an area where in the right project, it's certainly worthy of scientific exploration. So I have just a couple more minutes, and I want to make just a few comments and a pitch, but really talk about patient advocacy, because I think that's part of what Net Awareness Day, month, year is all about, and the opportunity that many of you have. Self-advocacy, looking at some of the specific definitions, it's the ability to seek, evaluate, and use information to promote one's own health. You probably all practice that when you see your physicians and your care team. Representing one's own interests within the healthcare decision-making process, and patient involvement in healthcare decision-making. This doesn't always come naturally to patients, and I think that you acquire the tools along the way, whether it's Net Vitals, to have everything written down, whether it is just your own process of self-education. But there are ways that you can use this self-advocacy and your knowledge and expertise, and most importantly, your experience, to have your voice heard in research. So there are a lot of different ways to participate, and there are many different types of research. There is the scientific research that we talked about in the laboratory or in clinical trials. There is health outcomes research, and that's the opportunity to really reflect your experience. So first of all, you can be educated yourself and your family, and educate others. We can certainly use all the help in the neuroendocrine tumor community. You can exercise your voice. INCA, which is an international coalition of patient groups right now, is running a SCAN survey that is open for another few weeks. Not only patients, but also caregivers and physicians are being asked to respond to that, to really talk about access issues and what are the barriers, what's working, what's not working. And again, this is international, so it's across the entire world. There's also issue-driven advocacy. I know that we certainly publicize those opportunities. LACNETS does as well. We did that a couple of weeks ago because there is a proposed change in Medicare reimbursement for the gallium-68 scans. So where you see the opportunity to comment or write to your representatives, should you choose, that's an opportunity to be involved and make sure your voice is heard. There are also some formalized patient advocate programs, and they vary with the organization, the agency, but the National Cancer Institute has a number of steering committees and task forces, and periodically they put out the call for people who represent rare diseases. There's also the FDA that has developed a patient-focused drug development process, and you can look on their website for that opportunity from time to time. PCORI is an organization that was funded by Congress, and it's the Patient-Centered Outcomes Research Institute. They are really funded on the philosophy that patients and patient experience has to be reflected in research. They get involved in a lot more of kind of health outcomes research, but again, it's worth a look at their website to look at how they craft the role of patient advocate. There's also NORD, which is the National Organization of Rare Diseases, and they do kind of local advocacy as well as education efforts from time to time. We're a NORD member as an organization. We've talked a lot about clinical trials today, so you definitely can look and see if there is a clinical trial for you. They're very specific, but I think sometimes if you have different physicians, if you're not seeing someone like Dr. Lee, who's just really so knowledgeable about clinical trials, make sure that your care team knows that you are interested in participating in clinical trial, and raise your hand and say that you're willing to entertain the possibility should something come along that is appropriate for you and where you are in your NET journey. The other opportunity that you all have that I want to make you aware of is you can donate some excess tumor tissue. And this is a project that we have been running. It's a collaboration of organizations. It's Net Research Foundation, an organization called Pattern.org, and we're working with a scientific institute in Boston, the Broad Institute. So the way that this works is if you are going in for surgery, they can take a little bit of excess tissue and they will actually FedEx it to the Broad, and the Broad will use its expertise and its resources to try and create cell lines. So it goes back to that research barrier of not having cell lines. Pattern.org is the organization that does all of the logistics. This is excess tissue that is not needed to determine your care. It is not exclusive of any tissue that you would donate to any tissue banks or any efforts here or at the facility where you are treated. It truly is excess. And it is an incredible resource and a way to be involved in research and perhaps help to create those cell lines. We've been doing this now with the Broad and Pattern.org probably for about the last 2 years. We've really stepped up our involvement and support over the last year. We're starting to see some good progress. Again, instead of trying to grow a cell in one medium or one, what I call, miracle grow, they're taking about 64 different approaches to growing those cells. So we think that it's a great project. We want to make sure that everybody is aware of this. I'd be happy to answer any questions, but you can go to pattern.org to do the consent online. And you can really make an impact in research if you're scheduled for surgery. So with that, I want to thank you for your attention. Again, it's great to be here. And Lisa, do we have some time for questions? Okay. Do you want to sit down? Or?
So we're running a little bit late, but what we'll do is we'll go ahead into our time of Q&A with Dr. Danley and Elyse Gellerman now, and then we'll take a quick stretch break. And at that time, the live stream will conclude, and then we'll have Dr. John Williams present for those of us in here, and then we'll break into our lunch. Okay. And I'll circulate with the microphone. for questions, but I just also wanted to, I want to thank both of Dr. Danley and Elyse Gellerman for this great overview on clinical trials and research. Just from a patient and caregiver perspective, I know there's a lot of information. So it's not that you have to know all the details. It's that we're giving the privilege of these wonderful people who are traveling around the country and around the world to all these medical conferences that you've heard them name. They're going all over, getting all of these bits of information and distilling it down as an overview for us. And that the big takeaway, especially for Neck Cancer Day, is Elyse said it so well, there's hope, right? There's a whole team of people working for us and with us, doing all this work out there. So we don't have to know all those minute details, but we know that this information is there and it's applicable to us. We have these videos available online. You can go back to it and look at it afterwards, but and then wherever it is that it relates to us. So they're getting a big overview with a lot of information. And as we look at it, then we can see, how does this apply to me? For my case, it might be my husband has pancreatic NET. So I might be looking at those specific trials, both for, looking at past data to inform my present decisions and also future decisions for other people that are living with lung NET. So it's not clinical. It's not just clinical trials as to like, what are my next steps, but what does the past data show that will inform my current decisions? And then also, what's around the corner for us. So thank you for that. That was really a lot of comprehensive, excellent information. And we'll take questions for either of them. Oh, and remember you're on live stream, so try to refrain from giving personal information.
I have had a neuroendocrine tumor mid-gut removed about 2 years ago. And when I had the gallium scan, they said I had lesions. What's the difference between a tumor and a lesion? Lesions in my bone and several other places.
So they would essentially, short answer is they're pretty much the same, because gallium imaging is very sensitive for neuroendocrine tumors, and usually you wouldn't have random spots that show up on the bone that's not necessarily related to your neuroendocrine tumor. So short answer is it's essentially the same.
Thank you both for being here. My question is relative to the status of one's cancer growth. If things are stabilized, does that still make you a good candidate for a research trial?
Mark, so I think that's a really good question. I think it depends on what the trial is. So, I think Elyse highlighted on that there's a lot of work in terms of kind of preclinically or correlative studies that are important to know to expand our knowledge and potentially look at other ways to kind of target the tumor. And then I really highlighted more on kind of the current therapeutic trials with active therapies. So it depends on the trial. So for the therapeutic trials, a lot of times as part of the eligibility criteria, it does require you to have disease growth prior to being enrolled on the clinical trial for most therapeutic trials. But as mentioned before, there could be, additional correlative studies for patients that have, stable disease, whether it's looking at tissue biomarkers, imaging biomarkers, whatnot, that you can participate in, or additional non-therapeutic studies that's looking at, patient preferences. We have an NIH grant that's a R03 that's looking at patient preferences right now that all patients with neuroendocrine tumors can participate in. So I think it really depends on that.
Other questions? Okay.
The gallium scan recommended for everyone who has endocrine tumor, or it depends on what kind of tumor you have?
Yeah, so the question is the gallium scan recommended for all patients, or does it depend on what type of tumor that you have? I would say for, kind of our community, and if you have a diagnosis of neuroendocrine tumor, then absolutely, I think a gallium scan is essential to get a sense of whether or not your disease express somatostatin, which is what a lot of our treatments are able to target, but then also to get a sense of how much disease that you might have, and potentially where the primary lesion is coming from. Yeah, yeah. So I would say if you have a diagnosis, I think that does warrant a gallium.
My question is, if a tumor is deemed inoperable and you have chemo and it's shrunk, is it considered then operable, or does it not, that might make a difference?
So the question is, if a tumor is initially deemed inoperable and you have some type of systemic treatment like chemotherapy and it shrinks, are you then a candidate potentially for surgery? I think that's again a really good question. That's a complex question. I think it also depends on which surgeon you're meeting, how aggressive they are, and what is the goal of surgical resection. It depends on how much the tumor has shrunk and what made you potentially an inoperable candidate to first begin with. If the issue is that the tumor is, let's say, close to vital blood vessels, for example, and if you have tumor shrinkage but it doesn't pull away from that vital blood vessel, then it still might not be appropriate candidate for surgery. However, if it's a situation where they were concerned about potentially clearing you of the disease because they were concerned about potentially leaving the disease, if you shrink it enough, then that might open the door. But if you have more extensive disease, let's say outside of the local area, then again, that's up for debate, and there's different ways to kind of think about how aggressive you want to be with surgical debulking.
One of the questions that came up online was how long you could keep taking an oral agent, if you could keep taking it as long as you could until it stops working.
Yeah, so for most of the oral agents that we have for neuroendocrine tumors, they tend to be, we recommend taking it until either progression of disease, meaning that your disease has developed resistance to that treatment, or taking it until significant toxicity where you're no longer able to tolerate the treatment at all.
If you have a functional tumor, can it become a non-functional or vice versa? Or is that, and if it spreads, it metastasizes, can the other places that it goes, can that be a different kind of functional or non-functional tumor?
Yeah, so the question is, if you have a functional neuroendocrine tumor, can it ultimately become a non-functional neuroendocrine tumor? And then depending on where the site of disease is, are there potential differences or something that's called tumor heterogeneity? I would say tumor heterogeneity does exist, meaning that certain types of tumors that's in your body for neuroendocrine tumors might behave slightly differently to others. Usually for a functional neuroendocrine tumor, it tends to stay, functional over time, unless I would say the caveat is, let's say it gets transformed into a much more high-grade disease, because oftentimes our really high-grade aggressive neuroendocrine carcinomas tend to be non-functional. And that's one case where that could happen. But I think your doctors should be monitoring your functional status, routinely throughout the treatment, because for functional tumors, remember our idea is it's not only important to control the tumor itself in terms of tumor bulk, but it's also very important to control the excess symptoms from the hormonal release. So you have to control the tumor bulk as well as hormone control.
So you touched on the fact that Medicare was changing on the gallium, how it's going to be reimbursing. Can you just elaborate on that for a minute?
Yeah, let me just give you a brief explanation. So when a new imaging agent was approved by Medicare, both the procedure and the agent were reimbursed. And that happens for a period of about 3 years. It's kind of what they do with new technology. Now we're getting to the point that Medicare is talking about rolling those two payments together, which usually in the final analysis ends up with a lower reimbursement. So that period of the initial 3-year reimbursement expires, I believe, in early 2020. And a number of societies, including the Society for Nuclear Medicine and Molecular Imaging, was trying to support legislation in Congress so that high-cost imaging agents like gallium would be reimbursed separately on an ongoing basis. So if you want to find out more, I can give you a couple of website recommendations. Josh Mailman in NorCal Carsonet has been very active in this area, and we put out some of this information as well. But really the request was that if you're concerned about this, please communicate with your representatives and try and really express support for what has become standard of care in our particular disease, because the fear is that if the reimbursement is less than the cost of the imaging agent, that the test may not be as available in as many places across the country.
As more and more people become diagnosed, is there any research with regards to genetic testing that should be done?
So that's also a really good question. While minority of neuroendocrine tumor patients where we've looked at potentially have a hereditary predisposition, majority of neuroendocrine tumors are what's known as idiopathic, meaning that we don't necessarily know what the root cause is. There is a lot of research in this area, not only to look at potentially additional genetic factors of risk. I think the NIH is specifically looking at that. There's a large study where they're trying to see patients, what their family histories are and everything, and trying to look at it from a deeper level from the genetic perspective. But then there's also, studies that are ongoing. I know our group specifically has an interest in this, as well as the UCSF group that presented some data looking at potential environmental factors. Because I've even encountered a few patients where there was not necessarily genetic predisposition, but first relatives having neuroendocrine tumors around the same age.
And I think I mentioned briefly in my talk that we do have an international team that's looking at a set of tumor samples, again, from multiple different perspectives, the genetics, environmental aspects, just to see if there are any commonalities. I think also in the book that we've given to all of you today, our NetRF neuroendocrine guide, there is a piece on genetics, and we get that question a lot at a lot of our programs. The first question is, if someone says, I have a neuroendocrine tumor, do my kids need to be tested? And it depends on what else may be present in your family. Is there anyone who is MEN1? Is there anyone who has one or two other rare genetic syndromes? So I think the best thing to do is really discuss this with your care team, and just be aware of any other illnesses in your family. Sometimes, you don't know what happened a generation or two back, but the best possible record and notes that you can compile, and then really have that discussion with your physicians.
When you have carcinoid of the heart, I know you could give your tumor tissue. When they do your valve replacement, are you able to give the valve or your heart to the study of NETs?
That's interesting, because we've actually not had someone ask that question, because we've been looking for excess tissue from intestine, lung, liver, pancreas. I don't know the answer to that, but I'll find out.
Singh has something to add.
The cardiac carcinoid is secondary, so it's never primary, and that's why there's a paucity of tissue. So that's why you're not going to have an immediate answer. It's very, very rare to have a primary neuroendocrine of the heart itself. Also, somebody asked a question about genetics. We actually looked at the California Cancer Registry 4 years ago to identify pockets of where these patients are coming from. So we were able to identify within California 7 pockets with very high incidence of neuroendocrine tumors. So we definitely think there's some kind of environmental thing going on with this. Within that, we acquired last year the food, water, soil, and pollution data. Problem for our statistician has been the way that is collected is completely not zip code based. So he spent over 1.5 years with no answers so far. But we're trying to identify if there's a single, something that's common that's happening. One of the pockets has been Central California, where there's a lot of farming done. So we think that there's probably some association with some of the could be, and again, it's completely hypothetical, so I can't say this on, with some of the pesticides or products that are being used over there. So, but there's a whole pocket coming from Central California too. It is not. It is not. Because there is no correlation. The pockets are actually, it's in one of the talks I gave for LACNETS about 4 years ago. So you may be able to see the graphs that I presented at that time. And then it kind of, when I saw these high incidence areas showing up, I said something's going on. And again, like I said, it's been extremely difficult even after we've acquired the data to find out or narrow down what may be triggering. And like Dan just mentioned, it's interesting because we're seeing people growing up in the same neighborhoods 20 years, husband and wives now having neuroendocrine tumors. And so we're seeing first-degree relatives coming up, but there's no genetic thing that we've actually been able to identify. We have patients now that are 18 and 19-year-olds, 16-year-olds that have had total pancreatectomy with liver metastases resections. We don't know what's going on. So there's something going on. We don't have a finger on it yet, though.
Thank you, Dr. Singh. And to see the past videos from meetings, you can go to our web page, our YouTube site is if you look at our LACNETS YouTube site, and under there you could search. So you can search Dr. Singh's name and then see the past videos. We can take a couple more if there's anything. And I just wanted to add, I mean, all this research stuff is so great, not just to inform our personal decisions and in our conversations with our specialists, but really for advocacy, dealing with insurance. I mean, when I know when we deal with insurance denials, I quote the data. You know, these are the trials, like that slide with all those trials, that's the data. That answers a lot of those questions that you might have of why am I even on the shot? Because the PROMID or CLARINET trials supports this. I mean, that's the reasoning. It's not just because Dr. Singh told me to take it, but because all of this study, all this work from all these doctors, all these institutions across the country and sometimes across the world, it supports you being on this treatment or taking these next steps. So any more questions? If not, we'll take a quick 10-minute stretch break, like 7-minute stretch break, and then like at 1 o'clock, we'll resume live for our next talk. And thank you for those of you who joined us on live stream, and we'll say goodbye to our live streamers.